All right, welcome. I hope everybody's doing all right today. We're going to talk about Chapter 4, which is all about structure and function of nucleic acids. And then at the end of the chapter, it speculates a little bit about where some of these large molecules that we've been talking about, the macromolecules, originated on early Earth, and speculates a little bit as well about how some of the first cells could have potentially originated. All right, so this is the fourth in our list of these groups of biologically important macromolecules. We already talked about proteins, carbohydrates, lipids, and so now we have nucleic acids. And nucleic acids are considered polymers, and they are built of monomers called nucleotides. And of course, there's two main types of nucleic acid. There's DNA, deoxyribonucleic acid, which we all know is the genetic material, and then RNA, ribonucleic acid. And we'll talk a little bit about the differences between these. Um, so each of these monomers, nucleotide monomers, really has three parts. There's a 5-carbon sugar, a phosphate group, and then a nitrogenous base. So looks something like this. So here's our one assembled nucleotide, right? Sugar in the blue, phosphate in the yellow, base in the orange here. And usually if you think of nucleotides, you probably think of the base, the nitrogenous base portion. So this is, the ATGC that we all know and love. So the bases are either pyrimidines, which have this single ring structure, or purines, which have this double ring structure. The pyrimidines are cytosine and thymine in DNA. In RNA, you'll see RNA molecules don't have thymine, instead they have uracil, and the purines are adenine and guanine. All right, so one of the really, aside from that one nitrogenous base difference, the other one of the other key differences between DNA and RNA is the sugar portion of the nucleotide. So RNA contains a 5-carbon sugar called ribose, whereas DNA has a 5-carbon sugar called deoxyribose. And as this name implies, really the only difference between these two is just this one missing oxygen in the deoxyribose, right? We deoxied the ribose, took away that oxygen. So we have that circular 5-carbon sugar. Um, and if we're going to take these monomers and start linking them together to build a polymer, um, we're going to have to, again, do some dehydration reactions and form covalent bonds. And again, we're going to see a special name for this covalent bond. It's called a phosphodiester bond, linking two nucleotides together. So here we're building a polymer. In this case, it's an RNA molecule, so we're taking RNA nucleotides. We know that's the case here because we have a uracil nitrogenous base. And so this would be one nucleotide here, right? Sugar, phosphate base. And we're going to covalently bond that with one of these phosphodiester linkages to the next nucleotide. So the bond is going to be between the phosphate group on one nucleotide and the sugar on another, right? And then we do that again, bond a third and a fourth, and so on. So we build what's called this sugar phosphate backbone of these polynucleotides, because we have alternating sugar, phosphate. Um, for any polynucleotide, DNA or RNA, um, the ends are chemically distinct. We talked about this being the case with amino acid chains as well. But in this case, the nomenclature is a little bit different. One end of a DNA or RNA molecule will be called the 3' end, and it always has this hydroxyl group just kind of hanging off the end there. Whereas the other end is called the 5' end, and it will have the phosphate group hanging free. So we build that polymer, and in the case of DNA, because DNA is double-stranded, we actually have to build two polymers and then stick those together. So two polynucleotide chains bonded together with hydrogen bonds between the nitrogenous bases. So you'll see we've got one polynucleotide here on the left, right? One sugar phosphate backbone with all the nitrogenous bases hanging off one side. And then we've got a second polynucleotide chain here, again with the nitrogenous bases all pointing off one side. And these two strands are oriented in opposite directions relative to each other, so they're considered to be anti-parallel to each other. And as I mentioned, then they're going to be stuck together with these hydrogen bonds, the little red dots you can see here, between the nitrogenous bases. And you may be aware of this if you've taken a biology class in the past, but we're always going to have what's called complementary base pairing. So if you have a thymine in this position on the strand on the left, you're going to have an adenine nucleotide in that position on the strand on the right. So adenine is always going to pair with thymine, cytosine is always going to pair with guanine, okay, without exception. So two complementary strands. RNA is typically single-stranded, although you can very often have instances like this, where this is one RNA molecule. You can see here's the 3' end, right? We could follow it all the way through to the 5' end here. So even though it is generally considered single-stranded, in one RNA molecule you may have complementary stretches of RNA that are capable of forming hydrogen bonds with each other, just like you would see here with the DNA. So you'll get this kind of unique structure where you'll have some portions that are truly single-stranded in gray here, and then some portions that are hydrogen bonded to each other in orange. Just a little table here summarizing some of those differences between DNA and RNA. So remember, we have different sugars, ribose versus deoxyribose. We have this one difference in the bases, the nitrogenous bases. RNA has uracil, while DNA has thymine. And then generally speaking, as I just mentioned, RNA is typically single-stranded, DNA is generally double-stranded. And as I'm sure you're aware, right, those two strands of DNA that are hydrogen bonded to each other are then twisted to form what's called a double helix, right? So it's a right-handed spiraling helix. And I'm sure you're aware of the idea that DNA is the genetic material and is storing information, and the way it does that is through the sequence of the nitrogenous bases. So we consider DNA an informational molecule. It is carrying the genetic code, and the code is the order of the nitrogenous bases in each individual DNA molecule. So in this case, if we're reading the left strand from top to bottom, right, the code, the information is TGAC, and that code is going to be different from individual organism to organism, and as we talked about in the very first chapter, right, this code is basically providing the information on how to build proteins in a cell. Now, I want to introduce this key idea, which is what's called the central dogma of molecular biology, which is all about how information flows within a cell. So this is arguably, the most important idea in modern biology, and it is the fact that information in a cell flows from DNA to RNA to protein. Okay, DNA to RNA to protein. And this is really a two-step process. So we make a RNA copy of our DNA through this process called transcription, and then we make a protein from that RNA through this process called translation. And you'll notice there's a little loop here on the DNA, and this is just implying that DNA can be replicated, so cells have to be able to copy the DNA. So that's the central dogma: DNA to RNA to protein, transcription followed by translation. And we're going to come back in some later chapters and talk about these processes in a little bit more detail. And if you take all the DNA in a particular organism, that is considered that organism's genome, right? So we can sequence genomes. We'll talk about that this semester as well. We can try to determine the order of all the bases in an organism's DNA, know its genome sequence. And another key term when you're talking about DNA is genes, right? You always hear about genes. So what exactly are genes? Well, genes are just portions of a larger chromosome, right? You can think of a chromosome as a large DNA molecule. I've got a video that I'll link out to here in a minute describing chromosomes in a little bit more detail. You can have this huge DNA molecule that is your chromosome, and small portions of that chromosome are going to be your genes. So for example, in this one DNA molecule, you've got a gene here, this whole segment of DNA. Then you've got a region of DNA here that is not considered a gene. And then you've got another segment over here that is again. And so it's just these bracketed portions, just these genes that actually contain the information to code for specific proteins in the cell. So we could transcribe gene 1 to make an RNA copy of that one particular gene and then use that to make protein 1, whatever that happens to be. And likewise, we could transcribe this gene over here, make an RNA copy of that gene, and then use that RNA to make protein 2 over here. So you have these genes scattered throughout your genome, but that's where a lot of the attention in biology gets paid to these gene sequences because we know how important proteins are, and so we want to understand, okay, what are these genes? What do they look like? How do they code for these proteins? All right, so as I just mentioned, I do have this video. It's kind of a nice description of chromosomes, so I will link out to that on Canvas. But I would recommend you take a look at that. And so, obviously, the most important role of nucleotides is arguably acting as that genetic material, acting as that blueprint for life. But there are some other very important molecules that are modified nucleotides. So for example, ATP, which I know you've heard of, right? That key energy molecule in cells, and its close relative GTP are actually modified nucleotides. There's some important signaling molecules like this one called cyclic AMP, or CAMP for short. We'll see that in an upcoming chapter. So just be aware there are nucleotides that are doing important things outside of DNA and RNA as well. All right, so at the end here, I just wanted to shift gears and just speculate a little bit about, where could some of these macromolecules that we've been talking about have come from in early Earth? And the bottom line is nobody really knows the answer to this, right? Because nobody was around to see it happen. But there is a whole field of science called chemical evolution. So there are people studying this. You can actually run experiments and try to test hypotheses and try to get a educated best guess on how some of these larger, more complex chemicals came about. And so all the evidence does seem to suggest that, things, life, even just chemicals started more simply and gradually gained complexity over time. And one of the early and more prominent experiments, I guess you could really say this kind of set off this whole field of chemical evolution, came from these guys Miller and Urey in the 1950s. Who tried to set up a in the lab simulation of Earth's early atmosphere. I'll just show you the image here. But their setup looks something like this, where they had an atmospheric compartment up here. So this is basically a closed system. So they had their atmosphere that contained gases in proportions that were believed to exist in the atmosphere of early Earth. Have an ocean compartment down here, basically just containing some salt water, right, that's being heated to give evaporation. So we're getting that moisture up into the atmospheric compartment. Got a condensation tube here to simulate rain. Um, and then we've got a couple of electrodes in here just providing some source of energy, right? Atmospheric energy, lightning, however you want to think of that. So they would run this as a closed system for a while, and then periodically they would collect a little bit of the condensation and the quote-unquote rain, and just look and see, what chemicals they were getting. Um, and interestingly, after running this for just a few days, they were actually seeing some amino acids being formed in this mixture, right? Some nitrogenous bases, purines and pyrimidines. So some of the either monomers or portions of monomers that could then be used to build some of these bigger macromolecules. Definitely seems at least plausible that they could have formed essentially spontaneously in Earth's early atmosphere. And so then if we think about taking these smaller pieces and putting those together to give some of these larger macromolecules, it seems likely that they probably would have formed on surfaces that would have had a very ordered, repetitive structure, almost serving as like a scaffold for building these repetitive molecules. So on the surface of things like minerals, for example, or crystals that would have very defined repetitive structures. And they almost certainly would have occurred in places where there was some heat energy, so near hydrothermal vents or hot pools, for example. So, again, we don't really know for sure, but we think some of these larger molecules probably could have formed essentially spontaneously on early Earth. My screen went black there for a second. Hopefully we're still recording. Another question that's interesting to think about is, in terms of the central dogma, which of these molecules came first? So DNA to RNA to protein. And it's tempting to say, well, proteins would have come first because they're carrying out all the important work in cells. But of course, it's a bit of a chicken and the egg kind of problem because, in all living cells, we need DNA to actually be able to produce proteins. Um, so then you might say, well, okay, DNA must have come first. And it's possible, but there's a bit of a catch there as well, because in order to like replicate DNA or carry out this process of transcription and translation to make proteins, we actually need proteins present to do all those things. So the DNA wouldn't really be able to do anything without the protein counterpart, which really leaves us with RNA. And this is something that I think most biologists agree on, is that RNA was probably the first genetic material and probably existed before DNA and proteins. And so, interestingly, there are RNA molecules that actually have some enzymatic activity like proteins do. These are called ribozymes. Um, you don't really have to worry about the details. These are basically just, as I said, RNA molecules that have some enzymatic activity. And as we saw earlier in this chapter, right, RNA is an informational molecule, can store genetic code just like DNA would. So it kind of bridges the gap between information and functional cellular activity. So it was likely an RNA world at some point. And then in terms of how the first cells might have originated, um, we talked about the fact that if you put phospholipids into an aqueous environment, they will spontaneously form a lipid bilayer. And in fact, they will actually spontaneously form little spheres. Some scientists will call protocells. So you can imagine situations where these little phospholipid spheres are trapping some of these other molecules that are being formed. concentrating them together, encouraging chemical reactions to happen inside the interior of that little lipid ball. So you could think of that essentially in its essence as, maybe the most primitive, earliest type of cell. But again, we don't really know for sure, but I think it's kind of cool to think about it and speculate a little bit. So anyway, thanks for hanging with me through that. And next time we will start in on Chapter 5, which is all about cells and different parts of cells.